Welcome to Tecan's latest webinar, Your Body is 3D, Why is Your Cell Culture Not? I'm Ashley, Managing Editor for Technology Networks, and along with my colleague Louise, I'll be moderating this event. Today's webinar will be given by Christian Oberdanner, Application Specialist Sales and Marketing for Tecan Austria. The presentation will also be given by Jens Kelm, CSO and co-founder of InSphero. With 3D cell-based assays increasingly important in life science, this webinar will explain the automation of a 3D cell culture and will discuss a long-term live readout of the latter with a microplate reader. 3D cell-based applications close the gap between conventional in vitro approaches and in vivo animal model research. Automating the 3D cell culture process and assay detection in a microplate-based format significantly shortens time of new drug candidates, as well as offering more reliable statistical results. Christian will present solutions for 3D cell culture systems using the Alvetex, Raft scaffold-based and Gravity scaffold-free technology. Jens' presentation will focus on the collaboration between InSphero and Tecan. Following the webinar, we'll have a Q&A session and welcome any questions that you may have. To ask a question, you should click the Ask a Question button to the right of the screen, enter your email address and question, and just click OK. We'll answer as many questions as possible during the time available. If you have any technical issues during the webinar, please also use this button to request support. So with this out of the way, I will now hand over to Christian for today's webinar.
So thanks very much, Ashley, for that very kind introduction, and a warm welcome to the audience also from my side. Together with my dear colleague Jens from InSphero, I will share some information and data about 3D cell culture today. So first of all, I will start with an introduction on 3D cell culture, explaining the benefits of 3D cell biology compared to conventional cell culture technologies, so 2D cell culture. Then I will quickly explain the major technologies on 3D cell biology, so how can we realize three-dimensional growth of cells. And then I will also talk about the activities of Tecan related to that very hot topic. After this, I will introduce two of our partners, which is Reinnervate, offering their Alvetex technology, and Biosystems with their Raft technology. And then in the second part, my colleague Jens will take over and will talk about his company, InSphero. He will explain the hanging drop technology which they offer, and then he will focus on the topic of differentiating drug effects in 3 microtissue cultures, focusing on applications performed on the Tecan's Infinity 200 Pro microplate reader. So conventional cell culture in 2D was used very successfully over the last decades. But we all know that the findings coming from conventional cell culture cannot always be directly translated to the in vivo situation. So there is still a huge amount of experiments that needs to be performed in element models. And this, of course, makes the whole process very cost-intense and time-consuming. So 3D cell culture can simply make the process much faster and at lower cost. This is simply due to the fact that the findings coming from studies performed in 3D are much more reliable, so the results are much more relevant and can be better translated into the in vivo situation, minimizing the amount of experiments performed in animal models. So what the researchers are looking for is what I already said, the greater biological relevance of the findings coming from 3D cell culture studies. There are also some other important factors or reasons why researchers look to the transition from 2D cell culture to 3D. which is, for instance, the enhanced cell viability and cell responsiveness, or a better quality of assay results. So going from 2D to 3D induces a lot of changes in cell differentiation, in cell metabolism, cell viability, cell proliferation, cell morphology, in the gene expression profile, but also in the drug metabolism. There is a huge amount of different publications available and a very nice comprehensive collection of this literature can be found on the homepage 3dcellculture.com. So the take-home message for the audience is that the results obtained with 3D cell culture are much more relevant. So how can you realize a three-dimensional growth of cells in culture? Basically, there are two major approaches. The first approach is the so-called scaffold-based approach, and as the name already says, these technologies use a scaffold to force the cells growing in 3D. This scaffold can be made out of natural or synthetic materials like nanofibers, hydrogel, polystyrene, or natural materials like extracellular matrix proteins, collagen, or laminin. The second approach, the scaffold-free technologies, include the hanging drop technologies, where microspheres are formed due to gravity and the cells grow in a drop. This will be in detail explained by Jens in the second part of this webinar. But also microfluidic systems belong to this category of scaffold-free systems. To show the compatibility to the most important technologies on the market, Tecan started collaborations with a range of companies focusing on 3D cell biology. This is first of all Reinnervate with the Alvetex scaffold system. So this is a scaffold-based system using polystyrene as a scaffold. A second company with which Tecan is collaborating with is Biosystems. They also offer a scaffold-based system called Raft. We started a collaboration with InSphero. They offer a scaffold-free system. More details will be presented later. And Tecan also started a collaboration with Celendis, and they are again offering a scaffold-based system using hydrogel as a matrix. So what are the exact activities with these partners? With Reinnervate, we basically tested the automation of their polystyrene-based Alvetex scaffold on our Freedom EVO liquid handling station. As you will see in the next slides, the process is pretty simple and very straightforward, and therefore the whole system is very reliable and very robust, resulting in a very high viability of the cells. With Biosystems and their collagen-based Raft system, we again looked on the automation of the technology on our Freedom EVO platform. This procedure is a little bit more tricky, as you need to mix the components first and need to optimize the cell growth. Furthermore, we also looked at the signal detection of signals coming from the Raft plate, and we optimized the readout with our Infinity 200 Pro multi-mode microplate reader. With Celendis, we again focused just on the automation of the technology on the Freedom EVO. And with InSphero, we more focused on the detection of signals coming from spheroids, and again used the Infinity 200 Pro multi-mode microplate reader. And you will hear a lot of exciting data in the second part of this webinar. So before handing over to Jens and before diving into the details of the collaboration with InSphero, I want to quickly present the facts about the collaboration with Reinnervate and with Biosystems. So basically with Reinnervate, as I said before, we focused on the automation of their 96-well scaffold Alvetex system. This approach or this procedure is pretty simple. You just need to seed the cells, add media, and then incubate the plate. So like you do more or less with a standard microplate in a 2D cell culture system. Due to the optimized gas exchange and nutrient supply of the scaffold, the cell growth is perfect, and the cell viability finally is very, very good. So this very straightforward approach results in a high level of standardization and robustness of the system. But how do the cells exactly grow in an Alvetex scaffold? And this you can see here on this slide. The cells are simply seeded on the scaffold and then will start growing into the voids of the scaffold, as you can see perfectly here in this cross section. Stained HapG2 cells, you can see here now, already grown into the void. Here you can still see some empty voids, and cells will grow in a couple of days in the whole scaffold, forming a real tissue, let's say. So with this system, you can perfectly mimic the in vivo situation, as you have a lot of cell-to-cell interaction, a very in vivo-like cell morphology, and as I already mentioned, a very good cell viability. I think this slide is more or less self-explaining. What you see here is a picture of a 2D cell culture compared to an Alvetex system, and you perfectly see the 3 dimensions of the cell culture, especially in the picture on the right side, and I think we all love this picture. It's really very nice. What we did when implementing the automation of the scaffold system on our Freedom EVO is we checked the cell viability compared to a manual handling of the plate. And what we finally saw is a typical growth curve absolutely comparable to the manual handling. So we found that the automation of the Alvetex scaffold is very straightforward, very easy, and results in the same or even better cell viability than manual handling. All these data are published in various papers available on the Tecan homepage as well as on the Reinnervate homepage. So the summary of the automation of the Alvetex scaffold. The Alvetex scaffold in 96 well format is easy to automate on our Freedom EVO platform. The cells are simply seeded on the top of the scaffold, and there is no other mixing procedure required. You can therefore increase the throughput dramatically, and this results then in a very high level of standardization and robustness, with a cell viability that is at least as good as with a manual procedure. Okay, so the second company I want to introduce, the second collaboration I want to talk about is with Biosystems. Again, we focused on the automation of the system with our Freedom EVO liquid handler. In this case, the whole procedure is a little bit more tricky, as you need to optimize the mixing of the components. The liquid handler needs to have some unique features like cooled carriers for gel preparation, a heating capability to induce the gelling, a BDK hood to keep the whole environment sterile. So not as easy as with the Alvetex system, but of course by automating the Raft technology you can achieve a very constant and reproducible scale preparation. But how does the Raft system really work? This is explained here on this slide. So you basically prepare a mixture of components that is then dispensed together with the cells into an empty well. You then induce the gelling at 37 degrees Celsius, therefore you need that heating option of your liquid handler. And then you add an absorber that starts to concentrate the gel into an in vivo-like collagen matrix. So finally, you have a tissue formed with collagen as matrix protein and the cells in between. This, of course, creates a very realistic natural environment for the cells, so that the cells can grow in multiple layers. And the nice thing is that you can analyze the cells with all the common technology used. So you can do imaging, you can do biochemical assays, you can do histological stains, whatever you want to do. And very excitingly, we found that the cell morphology, for instance, for dermal fibroblasts is totally different compared to 2D cell culture. So the Raft technology offers some special features. As I said, the analysis of the cells can be done very easily in a 96 well plate using a multi-mode microplate reader like we did with our Infinity M200 Pro. You can also do imaging, high content analysis in the 96 well format. You can even harvest again the cells using collagenase and centrifugation. So you can do something like flow cytometric analysis as well. And finally, the analytical plates are available in black or clear as it's needed for your detection. So again, we have published some papers and some posters available on the Tecan homepage and on the TAP Biosystems homepage. And the summary of the Raft technology, the automation of the Raft system in the 96 well format works perfectly on a Freedom EVO due to all the special features our liquid handler offers. We optimized the mixing of the components of collagen and cells, and this finally resulted in a very high level of standardization and robustness, also with the Raft technology. And again, like with Alvetex system, we could show that the cell viability is at least as good as with the manual procedure. So, thanks a lot. And with this, I want to hand over to Jens to share all the nice data that we generated together with InSphero. Thanks.
Thank you, Christian, for the nice introduction, and I appreciated the very constructive collaboration between Tecan and InSphero over the last 2 years. I think we have generated quite some decent data to help customers to implement the more complex and advanced 3D cell culture model systems into their routine drug discovery processes. I would like to talk a bit about how to differentiate drug effects in 3D microtissue model systems. Briefly to my CV, although Ashley already introduced us, so I have my PhD in biotechnology, where I already started to work on artificial microtissues. After my PhD, I've performed my postdoc at the University Hospital in Zurich, where we were evaluating whether we can use microtissues for cell-based therapies before becoming a scientific director of the Competence Center for Molecular medicine and applied biotechnology, where we have used various microtissue model systems for different applications in the medical sector. Since the beginning of 2010, I became the Chief Scientific Officer of InSphero. I'm also co-founder of InSphero. InSphero is a company based in Zurich in Switzerland, and we have been operational since January 2010, actually. So InSphero is working on model systems to increase the biological significance in vitro. We would like to reflect the in vivo tissue morphology and function more closely than it is currently possible with monolayer systems. And this shows here just an example of the heart. What we do not want to reflect is a whole organ. We would like to mimic the smallest functional tissue unit of an organ, as you see here, and not the whole organ. And on the right image, you see several different microtissues, myocardial microtissues, which are fully functional and for more than 2 to 3 weeks in culture, showing their functionality by contracting over time, and that's the functionality we would expect from a cardiomyocyte in vitro. However, a tissue is not only composed of a single cell type, which would make things on an analytical basis a bit easier. Tissues are composed of multiple cell types. And if you would like to discriminate effects on different cell populations within a tissue model system, we need quite specialized technologies to get the discrimination. The technology which InSphero is employing is based on the hanging drop technology. The hanging drop technology is one of the oldest technologies on the market. It's already 100 years old. Ross Granville Harrison, 1907, already used the hanging drops to grow frog neurons in vitro. The hanging drop technology is also quite popular in stem cell biology, where it is used to generate embryoid bodies, and it is the most versatile technologies to produce all different kinds of tissues, either disease models like multicellular tumor spheroids or primary tissue model systems like the liver, the heart, and so on. The basic concept of reforming microtissues in the hanging drop is quite simple. You prevent adhesion to a surface, the cells accumulate at the bottom of the drop, they start to assemble, they start to make cell-cell contacts, they start to make cell-type specific extracellular matrix, and that is very important that the cells make their own cell-type specific environment, because the environment in return determines the functionality of the tissue. And depending on the tissue type you are producing, after 4 days you have a spherical compact microtissue in your hanging drop. The hanging drop system allows a very precise definition of the initial cell number, so we can control the size of the tissue quite precisely, and that is important in 2 aspects. For one is you can control whether you would like to have hypoxic conditions or not. A size between 250 and 300 micrometer sustain norm oxia within the core of the tissue, whereas tissue of more than 400 micrometers in size have hypoxic conditions in the core with beginning necrosis. And depending on the model system, whether you would like to have a model system to assess safety over a long time, where you would like to avoid hypoxic necrosis, or you would like to design a tumor model system where you would like to have hypoxic necrosis because this reflects more closely the situation in vivo. And you can also perform co-culture systems. You can mix cells in the hanging drops, or you can preform a microtissue in the hanging drops and then add a second cell type into the droplet to generate co-culture systems. However, the hanging drop system is a very versatile technology to produce all different kinds of tissues, but when it's getting down to analysis, the hanging drop is not the best format to use. Therefore, we have complemented the production platform with a versatile analytical platform, the GravityPlus plate. Both plates are standard 96-well format, and you transfer the microtissues from the hanging job, as you see here, directly into the receiving well, which are placed below the production plate, and you just add excessive volume, excessive liquid of medium into the hanging job, and the job is falling down into the receiving well, and you can maintain the microtissues over time in this standard cultivation format. The GravityPlus is non-adhesively coated, so you do not get adherence of the microtissue to the surface, and you can maintain them over at least 4 weeks, 6 weeks in culture if you do regular medium exchange. The plate is also compatible with all different kinds of detection systems, such as the Tecan plate reader systems, where the big advantage is that the cultivation chamber is so small that with the detector of the Tecan multiplate reader, you always measure the signal of the full well with the system. You do not need to make any multi-spot measurements. Over here you can see a microtissue in the GravityPlus plate. This is the cultivation chamber, and within this cultivation chamber, which is 1 millimeter in size, you have the microtissue. And what is important is that you have a flat and planar bottom, which allows for good optical properties, so you can observe the microtissue with the microscope, or you can also implement the plate into a high content setup. The current golden standard of using cells for in vitro testing is still the monolayer cultures. Monolayer cultures have been used now since 40, 50 years in the labs. It is a highly controlled system. It's a very homogeneous cell population. Every cell sees the same oxygen concentration. Every cell sees the same nutrient concentration. There's no diffusion of the substrates required or of the compounds. However, we have also low to no production of extracellular matrix. However, even though it is a very controlled system, this is not what we have in the in vivo situation, as exemplified here for a tumor tissue. The tumor is a totally heterogeneous cell population, and we not only have different cell types in the tumor, also the cancer cell itself, they are highly heterogenic depending where they are located within the tumor, whether they are close to the capillary or far away from the capillary. They have different proliferation profiles. We have different nutrient gradients, oxygen gradients, metabolite gradients. So it's a completely mess, which is not reflected by the classical monolayer cultures, but which impacts the sensitivity towards drugs. This is much better reflected, the heterogeneity is much better reflected by the spheroid model system, where you do have all the different gradients like you would have in the tumor tissue. You have the oxygen gradient, proliferation gradient, nutrient gradient, and depending on the size of the spheroid, you get necrosis in the core of the tissue, reflecting the same heterogeneous cell population of the in vivo tumor. We have high production of cell type specific extracellular matrix, and we do have diffusion and diffusion resistant of compounds, but that reflects quite closely what happens also in vivo. However, a tumor does not consist only of tumor cells. A tumor consists also of stroma cells like fibroblasts and endothelial cells, and you can design your microtissue model system by increasing the complexity of the model. And this is an example here using the colorectal cancer cell and HCT116, where we have on the one hand we have the single cell type culture system, a very compact, dense system, stained here for epithelial growth factor receptor. However, then you can start to implement the different stroma cell components as shown here, where we have a co-culture system of HCT116 in combination with fibroblasts. You see the fibroblasts in the core of the cancer tissue shown by the Vimentin stain. And then you can add a third cell line. In this case, in the second example here, we have HCT116 and fibroblasts and endothelial cells, but we have stained only for the endothelial cells, as shown here for a CD31 stain. And you see that the cells are already starting to begin making capillary-like structures within the tissues. Two examples how the stroma environment can impact the morphology of the cancer cells. One example is MCF7. MCF7 is a breast carcinoma cell line, and if you grow them as a single cell type model system in the hanging drops, you get this micro tissues within the droplets, as shown here. The micro tissues, they have vacuoles indicating differentiation of the cells towards a more epithelial phenotype, and we have a high proliferative capacity of the cancer cells in the system. However, if we perform a co-culture system, as shown here, where we mix MCF7 in combination with the dermal fibroblasts, we get, first of all, a reorganization of these two different cell populations. We find the cancer cells at the outside, as shown here for the H&E stain, and the fibroblasts at the inside. And when we look at the Ki67 stain, which is an indicator of proliferation, we see that the cancer cell compartment is highly proliferative, whereas the connective tissue core is non-proliferative at all. And this is also a major difference of 2D and 3D model systems. Primary cells and non-tumorigenic cells do not proliferate in the microtissue model system. Only tumor cells and stem cells are still proliferative in the microtissue. When we compare now the morphology of the MCF7 in the co-culture system with the morphology in the xenograft system, we see that the co-culture system more closely resembles the morphology of the xenograft in vivo. One other example how the co-culture system can affect the receptor expression, the receptor expression of a model system is shown here for HepG2. We have stained for epithelial growth factor receptor, and there are a couple of clinical trials running where EGFR is a target for hepatocarcinoma therapy. However, when we grow HepG2, which is a hepatocarcinoma cell line, we see hardly any expression of EGFR in the model system. However, when we grow them with NIH53, a mouse fibroblast cell line, to reflect more closely the xenograft situation, which is also non-EGFR expressing cells, that's not surprising, it's a connective tissue cell. So when we do the co-culture system, where we implement the stroma cells into the model, we see that we have a highly upregulated EGFR expression of HepG2, which completely alters their responsiveness to compounds which are targeting EGFR. One application example where we have used the co-culture system for assessing combinatorial drug effects is shown here. We have done these tests in collaboration with Physiomics, which is a company in the UK, which have developed computational modeling algorithms to predict the effect of drug combinations. And together with them, we have developed a co-culture system with MX1, which is a breast cancer cell line, in combination with the stroma component with NIH3D3, the mouse cells, which I've shown before. And one of the most important characteristics of this model system is that we have been able to monitor growth over a long time period. And it's only the cancer cells which are growing over a long time, as it's shown here. So the connective tissue control shown in the blue, shown in the blue line over here, They do not grow over time, and when we do have the co-culture system with the tumor cells implemented, we see that they are significantly growing over time. And we need the long-term culture conditions to assess the combinatorial drug treatment, because also in vivo, it's not you do not measure size over 3 to 4 days. You measure growth of a xenograft over at least 1 month in the model system. What we have done initially is that we have assessed the range, the concentration range, which we would like to use for the combinatorial drug treatment, and this is shown in these two different graphs. This is shown in these two different graphs. So after 7 days, we haven't seen any impact of the compounds used in this study, gemcitabine and docetaxel. So you see here that even with high concentrations of 10 micromolar, we do not see any severe impact on the growth of these tissues. However, after 21 days, we see that with increasing concentrations, we can see an effect on growth of these tumors with increasing concentrations. So we use concentrations which do not have shown any effect after 21 days, and we have used concentrations which have been shown effect after 21 days to see the full range, and this is shown here, where we will see a clear combinatory effect of gemcitabine and docetaxel at very low concentrations. The single cell concentrations do not have any severe impact on growth. However, in combination, as shown here with the green bar, there is a clear decrease in size as compared to the controls. And the higher the concentrations are getting, the lesser the combinatorial drug effect is apparent. So how to discriminate now the drug effects in these more complex co-culture model systems? And there are two possibilities. One is that you're introducing fluorescent reporter systems, like shown here in this case, where we have used a cancer cell line, again the HCT116, tagged with the GFP with the green fluorescent protein, and the fibroblasts, which are harboring a red fluorescent protein. And this allows us, using the multiplate reader, to discriminate effects on these different cell populations by measuring the fluorescence intensity of the green channel and for the red channel. What was quite striking initially when we have measured the fluorescence intensity with the plate reader, we saw that the confluent monolayer cultures had a much lower fluorescence intensity than the microtissue cultures. And that is quite easily to understand because we have a much higher cell density at one place in the dish as compared to the monolayer cultures, where we have only a thin layer of cells distributed over the plastic. But this means also that we have a much better dynamic range for our assay window afterwards. So what we have done is we have compared the dose response using the green fluorescence channel, measuring the signal intensity of the GFP, measuring the signal intensity of RFP, which is reflecting the stroma compartment, and compare this to a classical LDH assay, which is reflecting the response of the overall tissue, including the cancer cells and the fibroblasts. And what you see here is that by measuring the intra-tissue LDH, we get an IC50 value of around 0.15 micromolar. However, when we look now at the different cell populations, which cell populations is contributing actually mostly to the IC50 value we have measured with the LDH release, with the intra-tissue LDH, we see that we get a good dose response measuring the GFP, so the cancer cells, they are eliminated with increasing concentrations of staurosporin. However, when we look at the RFP signal, there is no effect observed. We even get a higher signal intensity of the of the fibroblasts, and that can be explained quite easily because when we look at the bright field images, this is the DMSO control, we still here see the intact outer rim of cancer cells surrounding the connective tissue core, and with increasing concentrations up to 1 micromolar here, we see that the outer shell of cancer cells is completely removed, whereas the connective tissue core is still intact. And that can be discriminated by using the GFP and the RFP signal, which is reflecting exactly what we see here in the bright field images. You can perform also target validation in 3D microtissues using, again, fluorescently labeled cancer cells, as shown here. The system we have established is, again, a co-culture system where we used cancer cells harboring GFP and NIH-53 as a stroma component, again, to reflect more closely the xenograft situation. The transfection of the cancer cells with the SERA library takes place still in monolayer cultures. And after we have transfected the cancer cells, we mix these two different cell populations, the cancer cells and the stroma cells, in the hanging jobs and produce the microtissues. This gives us quite a wide variety of playing options because we can transfect only the cancer cells, we can transfect only the stroma cells, or we can do a combinatorial transfection to see how two different knockdowns in these two different cell populations are interacting with each other. Then we form the microtissues, which takes around 4 days, and after the 4 days we transfer the microtissues in the assay format, and then we do, again, a readout of the fluorescence intensity with the multiplate reader from Tecan. This is how the knockdown looks in the bright field images. What you see here is the mock transfected control after day 7, day 10, and day 14. You see nice growth of the tumor model system. In a similar way, the SEGFP knockdown control is growing over time. However, when we knock down KIF11, which is involved in the microtubuli formation during the cell cycle, we see no growth at all in the microtissue model system. And when we compare the knockdown efficiency or the knockdown effect in a monolayer culture compared to the microtissue culture, you see here that in the monolayer model system, there is no effect of KIF11 knockdown. It's still proliferating in a similar way than both controls, the GFP control and the untreated control. However, in the microtissue model system, we see that the cancer cells completely stop proliferating in the 3D model system. And there's one compound actually which is currently in the clinical trials, which is monastrol, which is targeting exactly the KIF11 pathway, the assembly of the microtubule formation. And this compound has been found by a phenotypic drug screening. However, using the monolayer system, you wouldn't have been able to detect this effect of KIF11. So if you would like to interact with InSphero, we provide different levels of interactions and products. InSphero is providing assay-ready microtissues, so we have a broad portfolio of different microtissue model systems. We have more than 100 tumor model systems in our portfolio. We have human liver microtissues, red liver microtissues for safety assessment. We are working at the moment on brain and heart. And so if there is anyone who is interested to become beta tester for red brain microtissues or for human heart microtissues, they can contact us. The assay-ready microtissues, you do not need to perform any cell culture by your own anymore. You receive the tissues, you put them for 1 day into the incubator, and the next day you can start with your assays. We also provide the 3D cell culture production and analysis platform, which allows a robust in-house production of microtissues, and we also provide screening and custom microtissue design services. So if you would like to screen a smaller number of compounds and you would like to assess whether the model system is of value for you, we can perform the assays and the screening in-house for our clients. With that, I would like to thank to the audience who clicked into this webinar, and I would like to hand over back to Christian for final summarizing this webinar. Thank you very much.
Thanks a lot, Jens, for that for sharing this very exciting data with us, and I'm already looking forward to the Q&A session. But before we start with this, I want to give a quick summary of what we have learned in this webinar. So first of all, Tecan offers a comprehensive portfolio for 3D cell biology. This includes various different 3D technologies, so we can handle the most important technologies on the market. And this ranges from the automation of the technologies up to the signal detection using our multi-mode microplate readers. Tecan really selected the partners with the most exciting technologies to give our customers the best solutions available. And all of these solutions offer true 3D cell culture. So if you want to have more information, you can easily visit our homepage, tecan.com. If you want to learn more about our detection portfolio, you see the links here on this slide as well. And we even have some target landing pages for 3D cell culture or for our gas control module, as well as for our hydrospeed plate washer that can, of course, also be used to, for instance, wash 3D cell culture plates. So thanks a lot, and now I will hand over to the Q&A session.
Okay, so thank you to Christian and Jens there for those very interesting and insightful talks. We have had some very good questions in today, I'm pleased to say, so I'll start off with question number 1. This is, are the Alvetex, Raft and 3D Life hydrogel technologies also suitable for dose-response experiments?
Okay, thank you for that very good question. And yes, of course, all of the 3 presented scaffold-based 3D cell culture technologies are suitable for dose-response experiments. So we have produced data with all 3 of them. We have published a poster with Reinnervate and Oncotest at the SLS 2013 this year. We also published their poster together with Celandis, and we will have an article with them in the JALA magazine, which was accepted recently. And also with TAP Biosystems, a nice application note is in preparation. So we see typical dose response curves, which indicates that the drug migrates into the scaffold and reaches and interacts with the cells as expected.
That's great, thank you. So the next question we've got here is, how can you be sure to measure the best signal in a 3D microsphere?
Okay, another very good question. As Jens already mentioned in his talk, the optical properties of the multi-mode reader perfectly fits to the properties of the gravity trap plate. And beside this, the set height optimization of our Infinity M200 Pro reader automatically looks at the set height of the microplate well, where the highest signal can be detected. So with this, we can always make sure to get the best signal from the microsphere.
That's great, thank you very much. The next one we've had in is in two parts. So to start off with, if you were to measure two different signals, for instance GFP, RFP, in a 3D co-culture system, how can this be set up in the software? And also, do you have to use special scripts or do you have to always intervene manually?
So maybe I will also comment on this, but I think also Jens can share his experiences with our multimode reader. So no, the reader itself and the eye control software allows to do a real multiplexing. That means that with a single measurement protocol, you can include all the important measurements or all the measurements needed. That means you can do an absorbance, a fluorescence or luminescence measurement all together in one single measurement protocol. You don't need to run multiple scripts or to interfere somehow manually. And maybe Jens, do you want to comment on this question as well?
Yeah, even though I'm not a specialist for programming of the multimode reader, we had very low difficulties to set up the multimode reader in a way that we can clearly discriminate between the GP signal and the RFP signal, which is in the case of these two different fluorochromes not as difficult because the wavelength of these two are quite far away from each other. But the bandwidth measurements of the multimode readers is quite precise, so that you can measure quite precisely without having any problems with crossover of each individual fluorochromes, different fluorescence intensities of different fluorochromes in the system.
That's great, thank you both for those. Right, the next question we have is what are the advantages of the hanging drop scaffold-free culturing versus the scaffold-based culturing, and do you have any comparative data for this?
I would like. I thought, Christian, that you might be the better one to kick off this question because I'm a bit biased. But in principle, the scaffold-free system has its advantages with respect to the development of cell-specific, cell contact, cell-specific extracellular matrix, so that you can reflect with this type of culture system more closely the in vivo situation than using either a collagen-1 gel, which is not apparent in all tissues in this amount, which is used for the Raft system, and also the polystyrene system. Has its limitations with respect to remodeling the in vivo-like extracellular matrix and environment. But again, I'm from InSphero, I'm biased here, and maybe Christian can a bit comment on this a bit more on a neutral basis. Sure,
of course. So as I tried to say in my part, Tecan looked at all the different technologies. Because from our perspective, let's say as a neutral party here in this area, we think that all the technologies have their benefits and also their drawbacks. And we really tried to select those who offer real 3D cell culture from our perspective. So from our perspective, there's not really a big difference. It's up to the customer to select the system that fits best to their requirements.
That's great, thank you very much. We have one more question that's just come in. This is one for InSphero. It just says, over the 21-day InSphero experiments shown for drug exposure, were the cells provided with fresh media, and if so, how?
Yeah, sure. So the long-term experiments, you have to renew the medium every 2 to 3 days. the medium exchange is performed in the same way as you would perform medium exchange in a regular 96-well plate format. Maybe just to clarify, we use the hang drop technology simply for formation of the tissue, and after we have generated the tissue, we are transforming the tissues to a non-adhesive specific 96-well spheroid plate, which we have designed, and the processes and the medium exchange processes are exactly the same. So you can do a regular medium exchange on a daily or every 2 days or every 3 days in the same way as you would do medium exchange in a regular plate format. And you can also do it on an automated basis, like with a Tecan automation system, to increase the throughput. It's absolutely compatible with all the automated handling systems on the market.
That's great, thank you. And I think the last one for today is one for Tecan. Can you control the plane of focus of the excitation beam for fluorescent reading in the Tecan multi-mode reader?
Yes, sure. So this is exactly our set height optimization function that we offer with our Infinity Reader series and with the Infinity M200 Pro. And we even included a nice unique feature, a signal to blank correction, so we can really find the set height, the plane of the excitation light where we get the highest signal but the lowest background.
That's great. Well, thank you both for answering those questions so clearly for us. We don't have any more in at this point, so all that's left for me to do is thank everybody involved in today's webinar. Thanks to the speakers, both Christian and Jans, for putting together the content, and of course thank you to all your attendees today for your time. We do hope that you found this Tecan series both enjoyable and informative. If for any reason you have missed any of the webinars, we're delighted to say that they will very soon be available through the cell culture community of the Technology Network's website. So thank you very much everybody, and hope you have a lovely weekend.